Good morning, viewers at home. My name is Bio-Edutainment Michael. I am here today to talk on microbial evolution. Microbial evolution. This is in furtherance of the educational series of Omega Bio-Edutainment. So, microbial evolution. And today, we are going to be so specific about microbial diversity and taxonomy. Microbial diversity and taxonomy. So from this word, diversity. So diversity actually means differences. Diverse, okay, that is in terms of, don't forget what we talk about evolution. I told you that evolution has actually made it very, very possible for us to be able to have new species, and that's why I said evolution is a very powerful tool for speciation. So don't forget that in the earlier video I told you about classification, that is, all living organisms are classified into 3 domains of life, and we also look at what is a phylogenetic tree. So any character or characteristics that tend to that two organisms or organisms share together, if they share it together, that is in terms of their similarities, and anyone that made them to be different. So it is now as a result of some organisms will be so similar in terms of their biochemical makeup, chemical makeup, as and so on and so forth. So, for example, when you look at Pseudomonas and E. coli, at some point in time, you just to a layman will believe that 2 of them are gram-negative organisms. They are gram-negative organisms. It is that gram reaction that made them to be grouped together, and that's why they are under the group Prokarya. They are under the group Prokarya. But evolutionarily, you will discover that, or biochemically, you just discover that these 2 microorganisms might not necessarily be the same in terms of their biochemical makeup or their biochemical composition, as well as enzymatic and some others. So, and that's the reason why we now have different types of microorganisms. Just like in human population as well, we just discovered that there are about 7 continents in Africa. If there are, I'm sorry, in the whole world. If there are 7 continents in the whole world, those that have the same boundaries, or they have the same boundary, the same color, they are grouped into the same group, maybe under the same continent. And those ones that they have diverse group are grouped under the. So that now makes the whole world to be filled with diversity, different types of diversity. Well, before we go on into then that is about diversity. Then we are going to look at taxonomy. Taxon means grouping. That is the art of grouping or classifying organisms into their various taxa, into their various groups. So we are going to see what evolution in microorganisms we have to look like in terms of their diversity and in terms of their taxonomy. So we are going to see this. And before we go on, I would like to introduce you into some basic terms and terminologies. In some basic terms and terminologies of microorganisms. Okay, now the first one is we are going to look at what do we mean by ecotype. What do we mean by ecotype? If you say taxonomy, what do we mean? G plus C contents. So what do we actually mean by this? And okay, CM, we are going to look at what this one means. We are going to look at what do we mean by a strain. Ecotype, taxonomy or taxon, G plus C contents, TM, as well as TM, that's a melting temperature. This is called melting temperature. Melting temperature. Okay, this is 1, this is 2, this is 3, this is 4, this is 5, and 6. We have also, what do we mean by systematics? Systematics. So please take note of all this. Now, and already I have told you what is a universal phylogenetic tree, universal phylogenetic tree. Okay, this is basically. So let's start with ecotype. Ecotype is a population of microorganisms that is genetically, so it's a type of microorganism, collection of microorganisms that is genetically different. They are genetically different, but they are in the same ecological niche. They are in the same ecological niche, and that is what is called ecotype. So that means if you now want to group some set of microorganisms based on their ecotype, that means you base it ecologically they are similar. So that means their ecological characteristics is what is going to make you to group them into the same group. But they are in the same group, however, genetically they are very, very different. They are diverse. So that is exactly what is called ecotype. That is genetically different and ecologically similar. So the next one is taxonomy. Just like I told you ever before, that taxon actually means the art. This is the science. Let me say the science of grouping organisms, not only microorganisms, of grouping organisms into their various taxa. And don't forget that I said taxa is into their various groups. So that is a taxonomy. Okay, so the next one is G plus C content. So this actually refers to as, don't forget that DNA is double-stranded and it contains, they are triply bonded into, G is triply bonded into C and A is double bonded into T. So the number, the percentage of the guanine cytosine content will actually make them to be what? To be, so the amount of G plus C content in a given organism is known as their G plus C content. And that is exactly what they use in classifying them into their various groups. There are some Microorganisms that, for example, if you now have G plus C content, let's say you have let's say you have microbes, so you want to group them into this. So let's say that means, let's say you have A, B, C, D, E. There are 5 microorganisms with different percentage of G plus C content. This is measured in percentage. If this one is 54.2, if this is 32.5, if this one is 67.8, if this one is 70.5, and if this one is 72.2. So if you are now to construct different types of, let me say, if you are to construct phylogenetic tree of organism A, B, C, D, E with this their G plus C content, you actually know that you are going to place E is going to be on your number 1. So followed by D, followed by C, then followed by A, and lastly it is going to be B. So what that means is that EDC, if you compare D and E, you will see that in terms of their G plus C content, this one has high G plus C content more than CAB. B has the lowest, followed by A, and followed by C. So if you are to group this type of organism, first of all, we are going to place high premium on D and E first. That is what brings them together. And the longer, the lower their G plus C content, the more diverse. The lower, okay, the lower their G plus C content, the further from the evolutionary tree. You have to know that. That is, the lower this one B with the lowest is too far, is too dissimilar in terms of their G plus C content, and so we are going to see all this as we actually go on. And the next one is melting TM, which is called the melting temperature. Melting temperature. Okay, the melting temperature. Now, let's say you have double-stranded DNA. You have double-stranded DNA. So, and don't forget that by the use of by the process called transcription, there will be separation of the strand. So this is going to be mRNA. So from that mRNA, we are going to have polypeptides. We are going to have polypeptides. So this exact temperature at which the double strand separates into single strand is what is called the melting temperature. It is what is called the melting temperature. So you need to actually know that that is the temperature at which the double-stranded DNA actually separates into single-strand DNA. That is that. Now, the next one is a strain. What do we mean by strain? A strain are descendants of a single a strain. Okay, a strain are the descendants of a single culture, from a single culture. For example, now let's say you first of all on day one you culture your microorganism. This is the colony. Culture your microorganism. This is the colony. So then this one you begin to subculture. So into when you are going to have the same, or let me say similar um similar morphology in terms of their morphology, in terms of culture, in terms of color, in terms of the appearance on coloring. So all these are called the descendants. These are the descendants of a single colony or a single culture. Okay, and that is called a strain. Now, the next one is systematics. The objective systematics is the practice. Systematics is the practice of assigning names. Is the practice of grouping, not only grouping, and assigning names into organisms. These are called systematics. The practice of grouping and assigning names into different microorganisms. So that is that. And the last one, which is a universal phylogenetic tree. Universal phylogenetic tree is the one that was propounded by Carl Woese, that living organisms are divided into three domains of life. So, and that is called universal phylogenetic tree. Okay. So, and you see that they are from the same roots, from the same roots. The first one is archaea. The first one is archaea, followed by prokarya, and followed by eukarya. Okay, so in this, and Woese was able to do this by the use of SSU ribosomal RNA data. So he collected data of single subunits ribosomal RNA, collected the data of different microorganisms, and he was able to come up with what is called universal phylogenetic tree by the use of SSU ribosomal RNA. So if somebody is asking you to write short notes on universal phylogenetic tree, you should be able to trace it back to the pronouncement of Carl Woese. At different times, I've actually made mention of this. So by the use of the use of universal phylogenetic tree, plotting of phylogenetic tree by the use of single subunits ribosomal RNA. So that is what is called universal phylogenetic tree. Now, the next thing is we have been able to look at, if you collect microorganisms from different sources, from different ecological niches, you will discover that at the end of the day, you are going to have different types of microorganisms that will behave in terms of biochemical makeup, in terms of their genetic makeup, in terms of the TM, that is the melting temperature, and you can also use G plus C content or percentage to actually classify or group them. So, and that is the reason why that by the time we start, you will see that there are some microorganisms that are called low G plus C, and there are some that are called high G plus C. So, based on this simple classification, we are going to see it. So, let's now go to assigning name, that is systematics or classification. What are the methods of classification of microorganism? What method can we use in classifying them? So the first one is called phonetic classification. Okay. Another one is the use of phylogenetic classification. That is, you use their phylogenetic information to be able to classify them. And the last one is genotypic. These are the very, very common one, genotypic classification. So PPG, that is a phonetic, phylogenetic, and a genotypic method of classification. So the first one, which is phonetic, this one is as old as the ages. It's as old as the ages because this is the type of classification that was basically based on physical or what you call morphological, the use of physical or morphological classification. If by now, if anybody is relying on phonetic method of classification, you just discover that you are going to erroneously classify organisms that are not related at all. You classify them to be in the same group. This was what Adam and Eve used in classification of organism when he was in the Garden of Eden. What he used was, organisms that fly and organisms that cannot fly. You will see that based on the physical or morphological characteristics classification, you just discover that you'll be able to, if you want to classify, you classify butterfly in the family of the birds. So, but you will know entirely that two of them are very, very distant in terms of their relatedness. And that is the use of phonetic. So this one is more a natural way of classifying organisms, So, and apart from the use of physical, some people also use motility, whether they can move or they cannot move. So that means if you want to classify microorganisms, those ones that have locomotory organelle, you classify them separately, and those ones that do not have, you classify them. So because you are only relying on phonetic classification, and if you are going to do that, that means there will not be any distinction between a parasite and bacteria. So, for example, if you classify two microorganisms A and B, if this one is a parasite and this one is a bacteria, now if this one is a Toxoplasma gondii, okay, and this one is just E. coli, because of the presence of locomotory organelle in these two, that means you classify them to belong to the same group. However, this one is a parasite, whereas this one is a bacteria. So that is one of the errors or the weaknesses of the use of a phonetic classification, and that was why scientists had to go for a more correct or more advanced classification. So, and that was when they now depend on the use of a phylogenetic tree. So the use of their phylogenetic tree and is classifying them. So for example, now if you draw a phylogenetic tree like this, if this is a phylogenetic tree, okay, now let's say you have, this is their roots, and this is A, if this is B, this is D, this is E, this is F, this is G, this is H, this is I, this is J, this is K, this is L, this is M, this is N, this is O. So if you are to use this kind of classification, the use of phyllo, don't forget that I said phyllo. Phyllo is actually talking about evolutionary relatedness and genetics. So the use of their genetic composition in terms of history, so in terms of evolution, we are using it to classify them. So if you now plot a phylogenetic tree to actually know whether the organism we are dealing with, how related are they and how distantly unrelated are they. For example, now you just discovered that A and B are more related than D and E. Then D and E are more related than F and G. All this. So that one is going to tell you the use of a phylogenetic tree in the classifying them. So just like what Carl Weisi used, he used SSU ribosomal RNA in classifying all organisms into 3 domains. So that is that. So then nowadays it is now very possible for us to go into, what you do nowadays is that you just go into the gene bank. That is where all genetic information of all organisms are being deposited throughout the world. So you go to gene bank, you get the sequence. You get sequence of this in terms of G plus C, A, and all this. You get that of all these organisms, that is from organism A to O. So by the time you are now comparing them, how related are they and how unrelated are they, that is going to help you to be able to plot phylogenetic tree and be able to know how distantly or how closely they are related. So that is so this is much more specific, and more accurate than the use of phonetic classification. So by the use of this, and nowadays they now have different software in bioinformatics to be able to plot a phylogenetic tree together. So the first you look at alignment, how related are they, how can you align them in terms of their similarities, and how can you separate them in terms of their distinctness or distinctiveness. Okay, now the next thing is to use the genotype. Okay, the next one is genotypic classification. That is when a scientist will now rely on the genotype of organisms in classifying them. Okay, so this actually come This actually came around the 1970. This existed until 1970 and it was very, very relevant until then. People rely basically on phylogenetic tree classification, phylogenetic classification, but they also find some weaknesses. They also find some weaknesses because there are sometimes that phylogenetic tree will also have its own limitation, and that was why they also would use or apply their genetic composition in classifying them. And anyone, okay, for example, now the use of genotypic, genotypic will tell you that anyone that is related in terms of 70% and above, they are similar. And anyone that is less than 70%, they are similar. So we are going to see this, and what they normally use nowadays is they use a program called BLAST. That is what is called basic local alignment sequence tools. Basic local alignment sequence tools. That is when you will now you will go and still go to the gene bank, go and collect different sequences, and anyone that, and you will now run what is called BLAST. You run BLAST. Okay, by the time you run the BLAST on the use of your computer, so when you run BLAST, anyone that has 70% relatedness will be termed to belong to the same group, but anyone below that will be too or distantly unrelated. So that is the use of genotypic classification in classifying microorganisms. Don't forget that I said different microorganisms would live in different habitats. They are diverse because as a result of evolution has actually made us to know, to have, because every day we have microorganisms conquering different niches. So, and how, before you be able to diagnose, before you be able to recognize them, you need to classify them based on their relatedness and based on how unrelated they are. Okay, so that is that about the different types of classification. 3 different types of classification. But the next thing is to look at methods, the methods or the techniques, the methods or techniques, methods or techniques. What are these techniques, laboratory techniques that you can use in classifying them? Techniques of classification or techniques of taxonomic. Let me be so specific. Taxonomic classification. What are these types? Okay, the first one is to rely on physiological characteristics. So what do we mean if we say physiological characteristics? That is talking about, for example, now there are some organisms that you classify them in terms of their biochemical makeup, that you say catalyst tests. When you perform catalyst tests, there are different types of, because based on the composition, the chemical composition of different microorganisms can be classified based on their behavior to catalyst tests. And what do we mean by catalyst test? That is ability to oxidize hydrogen peroxide into water and oxygen. There are different types of, let's say you have 20 microorganisms. You have 20 different types of microorganisms, and you perform catalyst tests on them, because those ones that will be catalyst tests means that they will contain an enzyme. They will contain an enzyme which is known as a catalase or peroxidase, that has the ability to degrade peroxide into water and oxygen. Whereas there are some that are catalase negative. So out of these 20, you just discover that maybe, just an assumption, let's say you have A, B, C, D, E, F, and by the time you perform catalyst test, you just discover that those ones that are catalase positive will be grouped together, and those ones that are catalase negative will be grouped together based on their physiological. Anytime you come across physiological, that is talking about functional, based on their functional characteristics. So that is physiological. Or this physiological is also the same thing as a metabolic characteristics in terms of the metabolic product. For example, there are some in terms of different types of glucose. There are some microorganisms that cannot metabolically undergo what is called glycolysis, because they do not contain enzyme that can break down glucose into pyruvic acid. So that will actually tell you that physiologically and metabolically you can use these characteristics to be able to classify microorganisms. So the next one is morphology. I told you morphological characteristics based on whether you can use that based on color, or whether there are microorganisms that by the time you grow them in the laboratory, you don't discover that when you touch them with wire loop, there are some that will be, in terms of color, some will be creamy, some will be whitish, some will be bluish in terms of color. And by the time you depend entirely on morphological characteristics to be able to classify your microorganism, it is going to be erroneous, because there are some organisms that they are all related, but they will exhibit the same color because they have pigments. They have pigments, and that is exactly what some media, for example, McConkey, Agar. So that one will differentiate microorganisms into lactose fermenters and non-lactose fermenters. Lactose fermenters into non-lactose fermenters. This is exactly going to be under metabolic characteristics. However, McConkey Agar has contained pigments, and you see them appearing in different color under this kind of agar. So that is that. Then there are some that will entirely depend on their biochemical behavior. We entirely depend on their biochemical behavior or biochemical characteristics. Example is fame. What is fame? That is the organism that can make use of fatty acid methyl ester. They contain biochemical that can actually that can break down fatty acid, fatty acid that contains methyl ester. So, and different types of these are very, very common. Now, let's now look at, let me try and go again over, I've mentioned morphological characteristics, I've mentioned physiological stroke metabolic characteristics, and I've also mentioned biochemical characteristics. Let me now look at, give you um, some of the characteristics that will fall under morphological characteristics. Number one, you look at the cell shape. Cell shape is under characteristics, morphological characteristics. Another one is the presence of cell wall. Presence of cell wall. So this one will actually tell you whether it is, there are some that they have thick cell wall and there are some that have very thick cell wall. You can actually use this in also classifying them. Then you can also use whether they have endospores, presence or absence of endospores. There are some microorganisms that at the end of the day they will have to produce what is called endospores. It's part of morphological characteristics. Apart from that, the presence or absence of flagella, that is whether they are motile or whether they are not motile. There are some that they are motile, but some that are motile contain flagella, whereas there are some that contain cilia, and there are some of them that contains villi. Okay. Then there are some, you can also make use of the colony color. Colonial color, I've told you that some of them they will appear creamy, some will be whitish, some will be bluish. These will fall under major morphological characteristics. Now let's now look at some of characteristics that will fall under physiological characteristics. Under physiological characteristics, stroke metabolic characteristics. The first one is whether they have the ability, okay, so this is a carbon or nitrogen sources. There are some that you classify them into whether they are chemotrophic, they are lithotrophic, and so on, whether they utilize carbon as their sole source of carbon dioxide. Then there are some that you make use of their energy sources. Where do they get their energy from? So this will fall under physiological characteristics. Then nutritional type. Just like I said, are they photosynthetic? Are they autotrophic? And what are they? Then I also told you that you can also make use of pH, their behavior, osmotic characteristics, or whether how do they actually perform in the presence or absence of some this. Then some of them will be whether are they able to tolerate salts? Are they able to tolerate salt or they are not? Then you look at their growth range and so on and so forth. Now that is so I've mentioned morphological characteristics, I've mentioned physiological and stroke metabolic characteristics, and I've also mentioned biochemical characteristics as well. I've mentioned that. Okay, so the next one to use, you can make use of their ecological relatedness. So don't forget, the first one is The first one is morphological characteristics. The second one is physiological and metabolic features or characteristics. The third one, is biochemical characteristics. Whereas the fourth one we are going to consider now is ecological characteristics. They can be classified, microorganisms can be classified into their various group or taxon by the use of their ecological behavior, ecological characteristics. Okay, so this will depend entirely on how do they respond to pH, how do they respond to oxygen and osmotic pressure. So that is under ecological. So the most recent and the most one now is molecular properties or molecular characteristics. So, and these molecular characteristics now has different types under that. So we have so the first one under molecular, that is the types of molecules do they have. Okay, so the first one is the use of DNA hybridization. The use of DNA hybridization. What do we mean by DNA hybridization? Okay, so this is the ability. For example, if you have let's say this is the community of microorganisms. This community of microorganisms, and in this you have A, you have B, you have C, you have D, you have E, you have F. Now, by the time you collect their DNA, so by the time you collect their DNA, okay, let's say this is A, let's say this is B, C, D, E. If you want to know how related A is to B, is to C, into D, into E, by the use of their molecular characteristics, especially the use of a DNA hybridization, what you will do is you will subject the this DNA into high temperature. You increase the temperature. So because whenever you increase the temperature, it is believed that the DNA will unwind. They will separate. Okay. So, and by the time they separate, the first thing is you increase the temperature. That is the Tm. Don't forget that I said Tm is the melting temperature. That is the temperature at which the double strand of DNA is unwinded. So when you increase the temperature and you now look at the Tm, you now look at the temperature at this point in time. Okay, so then the next thing is you decrease. You allow it to cool, decrease the temperature. By the time you decrease the temperature, once the temperature is already going down, the unwinded DNA will begin to combine again, to align together randomly. And at this point in time, you can have anyone that will bind most. We have bind most. Those anyone that has, if they are similar, that means, so what you will do is that you are going to radioactively label A, B, C, the strand of this A, B, C, D, E. You will radioactively label them before you increase the temperature, or you can add fluorescent dyes on them. And by the time you added fluorescent dyes to radioactively label them, and you now increase the temperature, this one will now unwind. And by the time you now decrease the temperature again, the DNA will now hybridize again. And by the time you now hybridize them again, you will see that there are some that will contain strand A here. The complementary strand can be from any of these. So, and that is what is called DNA hybridization. So we are going to see this when we actually go on. That is a DNA hybridization. Okay. Then the next one is a nucleic acid composition. Nucleic acid composition. There's a formula to use. That is molecular percentage is equal to G plus C into G plus C all over A plus T, then G plus C multiplied by 100%. So the molecular G plus C content is the number of guanine plus cytosine and adenine and thymine. So you add them together. Let's say so you can actually use this to be able to do what? To classify microorganisms based on their relatedness and unrelatedness. So, and this is a typical examples of what we are going to see. By the time we go on. So, for example, you could be given different types of different numbers. They can give you like 20 bacterial species. Can be given 20 different bacterial species, and they will give you their G plus C contents, or they can just give you, they may ask you to calculate the percentage of G plus C. This is the formula you are going to use. That is G plus C is equal to G plus C all over number of T. You should know that the number of A will be equal to T. The number of G will be equal to C. You should know that according to charger flow of DNA. I'm sure that whenever you are doing biochemistry of microbial biochemistry, you must have actually done with the structure and composition of DNA. So don't forget what we have learned today. We have learned today what is diversity. What what is diversity, and what do we mean by taxonomy, which is the science of classifying organisms into their various taxa. Okay. After that, I highlighted like 6 or 7 different different um, terms and terminologies there. We look at what is an ecotype, we look at G plus C content, we look at melting temperature, which is TM, we look at what is universal phylogenetic tree, we look at what do we mean by systematics, we look at what do we actually mean by others. Then we proceeded to actually learn that how are organisms being classified? How can you classify them into their various groups? So we said, I mentioned 3 for you. The first one is called phonetic classification. That is that type of classification that is based on physical appearance or morphological appearance, whether they have locomotory organelles or not. So that is based on phonetic. Then we also look at a classification that is based on a phylogenetic tree, the use of phylogenetic tree, and I told you how related and unrelated microorganisms could be. And lastly, we also examine classification that is based on the genetic composition of different microorganisms. So before we now started with what are the basic techniques, laboratory techniques that you can use in classifying microorganisms. And don't forget that don't forget that I told you this is very, very applicable in the diagnosis, and correct treatment. If you cannot diagnose well, if you cannot diagnose correctly, you will definitely mistreat. So this method is what they use in diagnostic microbiology. Don't forget that if you are going to diagnose, that means you should be able to isolate them first, and not only isolate, you should also be able to identify. By the time you isolate and you are able to identify them, that's when you can actually determine the type of microorganism that you are actually talking about. I told you about some of the errors or weaknesses of phonetic classification, and where I mentioned E. coli, and tags Toxoplasma gondii. So Toxoplasma gondii is a parasite, whereas this one is a bacteria. Although 2 of them, they have locomotory organelle, that is, 2 of them can move. So if you only depend on locomotory organelle, that is the use of phonetic classification, in classifying them, so you will have erroneously group these 2 to be in the same group, whereas they are too distant. So we consider morphological characteristics, physiological, and I listed out some of the characteristics that you can classify to be under morphology. And I also mentioned that of physiological and metabolic characteristics, mentioned that of give us example of a biochemical classification, ecological classification, and the last which we are considering now is molecular characteristics. And I told you that the first one is how can you hybridize their DNA, the way they hybridize, or the degree of their relatedness. The degree of their relatedness will hybridize their DNA the more. So if the 2 organisms that are related, their DNA will hybridize more than organisms that are not related at all. And I also mentioned the use of nucleic acid composition. That is the use of G plus C content in be able to actually determine whether they are related or whether they are not related. So we still have more types of more different method of classification of classifying microorganisms, and we are going to see that, for example, after nucleic acid composition, we have nucleic acid sequencing. Nucleic acid sequencing, such as DNA, RNA sequencing techniques. We look at genomic fingerprinting and some other. So let us just stop here today. In the next video, the next time we are going to come online, I am going to actually continue from where we are stopping today. Once again, I want to sincerely